Welcome to My Long Island TV. From Manhasset to Montauk, we have traveled our communities to bring you the following events. I'm your host, Waldo Cabrera. My Long Island TV starts now.
What I'd like to do is we're going to announce names, and we want you to come up here, and we want you to very briefly describe your project, and here's why we're doing this. Because I can't even pronounce some of the projects that you guys perform, so we're going to have you do it.
I researched the most environmentally effective way of cleaning up an oil spill.
I studied proteins that are involved in gene regulation with the goal of better understanding development, and I specifically identified 2 novel factors that are involved in that process.
My study focused on alternative splicing and its effects in breast cancer. So basically analyzing the changes in gene regulation and transcription and ultimately genetics and how it affects the changes in breast cancer.
The focus of my research was to synthesize a compound that would not become resistant as traditional antibiotics have. So we were successful in not only synthesizing the compound, but also identifying where precisely this compound binds in the pathogen that causes TB.
And I was able to identify a number of gene targets that were responsible for regulating the development of the brain. So these gene targets might serve as potential therapies for patients of visual impairments, including amblyopia.
My study was on spinal muscular atrophy, which is the leading genetic cause in infant death.
My project was on studying the racial disparity in prostate cancer, and I hope to identify what factors or pathways are different between 2 races, African Americans and Caucasians.
I'm working on patenting to basically expedite and make the process of diagnosing mammograms more efficient.
I conducted my project on black holes and particle emission from them. I want to see if they can be detected today by NuSTAR, NASA's space-based X-ray telescope.
I worked on with a chemical analysis technique called laser-induced breakdown spectroscopy.
So my project was on a novel signaling pathway that I identified in cancer, in the majority of breast cancers that is dysregulated. And I found that inhibiting the signaling pathway could overcome resistance that's seen in breast cancer today.
My project was on a nonverbal behavior called power posing. So basically that entails that by standing in the open, expansive position, you actually can make yourself feel more confident by increasing your testosterone levels.
I studied this reaction within our blood vessels called the nitric oxide deoxygenase reaction. And I guess I used a novel way to study this reaction, and its future implications include solving complications during blood transfusions and blood substitutes.
So my project was a bioengineering project that focused on developing an ideal biomaterial that could use nanotopography to engineer a cell response.
I determined that lysine barbarum may act as a potential repair mechanism and protective one for Alzheimer's disease.
I wrote a simulation for the NuSTAR Observatory, like Caitlin, but to measure high-energy X-rays coming from the auroras at Jupiter.
I developed a conductive and resilient polymer gel that can be used in a future system that I am developing right now that can treat mitochondrial diseases in the future, including Alzheimer's, Parkinson's, diabetes, and cancer.
In my project, I studied the structure of the human organ recognition complex, or the protein that initiates the process of DNA replication in humans, and I managed to produce the first 3-dimensional model of the protein.
So basically, I designed a novel implant system to use the spine's natural growth to treat congenital scoliosis.
My study investigated the correlation between energy drink consumption and the use of some potentially addictive substances, including alcohol, marijuana, nicotine, prescription drugs.
Now do you know why I'm so optimistic about the future? Give them all a big hand.